Hi everyone, and welcome to Chapter 3, Biological Macromolecules. And remember that we learned in Chapter 2 that term macromolecule just means big or large molecule. And so that's what we're talking about in this chapter, which there's a lot going on here. So I've divided this presentation into two parts. So your objectives for this chapter are to be able to list and describe the four types of macromolecules in terms of their subunits, their functions, and be able to give examples of each of the four types. Explain how macromolecules are assembled and disassembled, and this is very important because these processes will carry through for the rest of the semester. Describe and or explain how the atoms and bonds discussed in Chapter 2 relate to the macromolecules in Chapter 3. And that's to remind you what I said back in Chapter 1, that everything is connected together. Everything in the natural world is connected together, and everything is built upon a foundation. And that foundation begins with atoms, and then we get into elements and molecules and macromolecules into cells into tissues. See how everything is built upon everything that came before it. And so the atoms and bonds that we discussed in Chapter 2 obviously going to carry over to macromolecules in Chapter 3. So, what is a macromolecule? You already know this. By definition, a macromolecule is a large molecule. We call them polymers. Polymers are long chains of something. So there you go. There's a good example. If you have a chain, right? If you have one link in a chain, you can hold it in your hand, and that's just one link. But if you have a 10-foot chain made up of those many links, okay, that's kind of like that's a good example, a good analogy for a polymer. A polymer is a long chain of individual links, and those links in the case of macromolecules or molecules are called monomers. Mono means one, so a monomer subunit. If you have many of these monomer subunits, they're like the individual links in a chain. You put them all together and you have a polymer. Poly means many. So it doesn't matter what macromolecule we're talking about throughout this chapter, and we'll see carbohydrates and proteins and lipids and nucleic acids, but they are all polymers composed of very specific monomers. And so one of your tasks in this chapter is to not only learn the name and the function of the polymer, but know what the monomer is and understand how the monomers are put together to make these big long chains. So macromolecules include the major types of organic molecules important to living systems, and these are the ones that we want to focus on. The names are not unfamiliar to most of you anyway. So carbohydrates, and you guys know carbohydrates as Snicker bars and whatever like that, right? So we have carbohydrates. Carbohydrates include, now this is a generic term, umbrella term, but carbohydrates in the natural world consist of, for instance, starch, glycogen, cellulose, and chitin, and we'll differentiate these in a little bit. We have proteins, and you guys think of when I say protein, you probably think of hamburgers, right? But proteins include enzymes. Enzymes are functional proteins, and then we have structural proteins, or proteins that are used to build things, to build other organic structures. And examples of structural proteins are your hair and silk from silkworms, the webbing from spiders, those kinds of things, okay? We're really going to stress heavily the enzyme function of proteins in this chapter. Lipids, that's a sort of a word that the lay public uses for fat. Lipid and fat kind of are interchangeable, but this includes fats and oils. We call those triglycerides because they're composed of, they have 3 subunits, fats and oils. And we'll also talk extensively about phospholipids. Phospholipids are those lipids that heavily compose the cell membrane or the plasma membrane. Okay, so which we obviously need to learn about, so when we go to the next step, cells, we'll have that understanding. And also steroids. Steroids are hormones, or there are different types of hormones, but steroidal hormones are also lipids. And then we have nucleic acids. Nucleic acids are, you're familiar with DNA and RNA, and we'll look at those structures in this chapter also. All right, so here's a nice table from another source, but it kind of breaks everything down for you, presents it at face value. Here are, let me get my little pointer. Here are your macromolecules, carbohydrates, nucleic acids, proteins, and lipids, which we'll learn extensively about in this chapter. And it gives you examples over here: carbohydrates, starch and glycogen, cellulose, chitin. But this is really important here in that it shows you or tells you what the monomer subunit is. So these are your polymers, and over here it gives you your monomer subunit. So for carbohydrates, we think of sugar, and so the subunit for carbohydrate is glucose. Glucose is, you might know that as blood sugar, right? That's the kind of sugar when you prick your finger and you take the little sample and you examine it for, that's for blood sugar, for the amount of sugar in your blood. That's glucose that you're looking for. Okay, so glucose is the subunit, monomer subunit for the polymer carbohydrate. Examples are starch and glycogen, and these are used for energy storage. You've heard the terminology anyway, when marathon runners, the night before they carb load, right? They eat pizza and lots of bread and pasta to build up their blood glucose, so they have lots of energy when they go out running 26.2 miles. Starch, the example they give over here is potatoes. Potatoes, yes, are lots of starch. In fact, only starch and water. But anyway, and glycogen. Glycogen is the storage form of glucose for vertebrate animals, okay? Cellulose is a structural carbohydrate, also made of glucose, subunit. Cellulose is structural support in plant cells, which we'll learn about later on. And chitin, it says modified glucose, but for our purposes here, it's glucose. Chitin makes up, it's structural, makes up the shells of crabs and lobsters and shrimp and that kind of thing. We have nucleic acids here, nucleic acids. The subunit is the nucleotide. The nucleic acids that we're concerned about are DNA and RNA. And this is obviously for carrying the genetic information. Proteins, the subunit for proteins is the amino acid. And you have functional proteins and you have structural proteins, and functional proteins are like catalysts, enzymes we call them, and for transport in the case of hemoglobin. and also for support, hair and silk, which I'll point that out again in a minute. So, but the important thing here for proteins is the structural, the monomer subunit is the amino acid. Proteins are nothing but big, long chains of amino acids. And we'll see that as we get into this chapter. And then we have lipids. Lipids are polymers of glycerol, fatty acids. Here we have triglycerides. Triglycerides because there are 3 fatty acids. They're used. Fats are used for energy storage. Examples here: butter, corn oil, soap. But lipids, we tend to think fats and oils. Okay, and then we'll talk extensively about phospholipids. Phospholipids, we have that little glycerol molecule, 2 fatty acid chains, and a phosphate group. Phospholipids are important components of cell membranes or plasma membranes, which we'll see and we'll look extensively at, and that will carry over into the next unit when we talk about cells, because we'll talk about the cell membrane and how phospholipids make up the cell membrane. Alright, let's see. Hold on. Alright. Pause. Okay, as we get into our study of macromolecules, you want to familiarize yourselves with functional groups. So functional groups of atoms, when attached to a molecule, they define the chemical properties and subsequently the functions of those molecules. And so you can see over here the various functional groups pictured here are found in carbohydrates and lipids, proteins, and nucleic acids, and also ATP. So I wouldn't so much say you need to sit down and memorize these like this, but should have a general idea because we'll see these again and again. So a hydroxyl group is an oxygen bound to a hydrogen, or I should say a hydrogen bound to an oxygen, and then that group, we call it a hydroxyl group, can be bound to carbon and other things. And we'll see this as we start synthesizing carbohydrates. The carbonyl group is this carbon. Remember, carbon can have 4 bonds. In this case, we have 2 single bonds and a double bonded oxygen. So 1, 2, 3, 4, those are your 4 bonds for carbon. So oxygen up here obviously can only have 2 bonds. But the point here is with carbonyl, it's a double bonded oxygen. Carbon double bonded to oxygen. Remember, those are covalent bonds. We just call it a double bond. You find those in lipids. So that's the carbonyl group. Here's the carboxyl group. Kind of like the carbonyl plus the hydroxyl group equals the carboxyl group, because here you have a double bonded oxygen to carbon and also a hydroxyl group bound to carbon. And we see this in proteins. All right, here we have the amino group. Now, amino refers to the presence of nitrogen. So here we have nitrogen bound to 2 hydrogen atoms. In this case, that's your amino group. We see those in proteins as well. And then your phosphate group, here is a phosphate molecule in the middle bound to 4 oxygens. That's the phosphate group. We'll see that in DNA and also in ATP. That's an important element of ATP because that's where the energy, ATP being the molecule of energy, but that's where energy comes from. When you knock off a phosphate group from ATP, then that releases energy and then you can do work. Okay, so Here's the basic structure of amino acids. Now we're gonna talk about amino acids, but I don't want you to go sit down and try and memorize all of these structures. That's not important for our purpose here. But the basic structure of an amino acid is important for you to understand. Okay, so here we have this carbon in the middle, bound to a hydrogen on one side. Remember, carbon can have 4 bonds, so it's bound to a hydrogen on one end or one side, and an R group. That's the functional group. Okay? And it varies from amino acid to amino acid. So the point here is with this amino acid structure is that all amino acids have this basic structure, and the difference comes in the 21 amino acids, right? The difference comes in the R group, right? That's the variable group. But every amino acid has this basic structure. So we have a carbon bound to a carboxyl group on one side. It's important for you to remember that, and an amino group on the other side. And as you can see here on the amino side, we have a net positive charge, and on the carboxyl side, you can barely see it here, we have a net negative charge. So that gives the amino acid a polarity. And that's important when we start building proteins, because you're going to have, remember, opposites attract. So you're going to have the amino group of one amino acid binding to the carboxyl group of an adjacent amino acid. Plus binds to minus, right? Positive to negative. That's the way that goes. So it's important that you remember this basic structure of an amino acid with its amino group positive on one side, its carboxyl group negative on the other side, and that makes the amino acid, gives the amino acid polarity. Okay, so a chain of amino acids, we call it a polypeptide because the bonds that hold amino acids together are called peptide bonds. And so we have this long chain, and you can see here's the basic structure that we saw on the previous slide, right? So here we go. So here's your carboxyl group on one end and your amino group on the other. Here's your carboxyl group on one end and the amino group on the other. We go down here, here's your carboxyl group on one end and the amino group on the other. Right? So we have this negative group here, that's the carboxyl, binding to this positive amino group. And this bond right here is called a peptide bond. And because this is a long chain, there are many peptide bonds, we call it a polypeptide. And this is the basic structure, we call it the primary structure of proteins. Remember I said earlier, proteins are just big long chains of amino acids. This is how we get there. Take an amino acid, link it up to another amino acid via a peptide bond, link that pair, that dipeptide, up to another amino acid via a peptide bond. Now you have a tripeptide. You link it up to another amino acid via a peptide bond. And now you have four. Once you have four or more, we just call it a polypeptide. And this can go on and on and on for hundreds or thousands of amino acids in this big long chain. Important to remember, the bonds are called peptide bonds, and polypeptides are always polar, with an amino group on one end. That's positive. We call it the N-terminus. And the carboxyl group with its little negative charge on the other end, we call it the C-terminus. All right, so the most important organic reactions, and the ones that we're going to be using the terminology, and we're going to be looking at these two reactions over and over and over and over again, okay? You're going to get sick of them, and that's what I want. So the first one is the dehydration synthesis. Now, without even looking at the definition here, think about what's going on. Okay, synthesis means you're synthesizing something, you're making something, you're building something. Dehydration, what happens when you get dehydrated? Why does that happen? Well, you sweat a lot and you lose water. So dehydration synthesis, by definition, we're going to synthesize, or we're going to make something by removing water, by dehydrating it. Another term here is condensation, which I don't use much. Molecules are joined together with a concurrent loss of water. That's the dehydration part. Molecules are joined together. That's the synthesis part. And so my little generic example here, A plus B yields the molecule that you're trying to make, all right, the synthesis thing, C plus water. We know it as H2O. The converse of the dehydration synthesis, here we're making something by removing water, is hydrolysis. Hydrolysis, or for emphasis, you will hear me say hydrolysis, because hydro pertains to water and lysis means to break apart. So we think of cell lysis, for instance. To lyse a cell means to break it apart, or to destroy it, or to rip it open. So here we're going to lyse something. We're going to break something with the force of water. That's the hydro part. So where we build something by taking away water here, dehydration synthesis, here we're going to tear something apart by adding water. So covalent bonds are broken by adding water. So here we go. Our little molecule up here that we made. We're going to take it, we're going to add water back to it, and guess what? We're going to break it apart and get our original reactants A and B back. Okay, really important for you to remember the most important reactions in organic systems. Dehydration, synthesis. You're going to make something by dehydrating, by taking water out. Hydrolysis, you're going to break something apart by adding water to it. And so here we go. Nice little image of you, for you, of dehydration and hydrolysis. So here we go. So here is this molecule here, and this little little molecule here. And in order to, so you see you have an OH on one side and an H and an OH on one side and an H. You know water is H2O. So in order to put these two together, we need to get rid of a water because we're synthesizing. And so to get rid of water, we pull an OH off of one and the adjacent H off of the other. Those two join together, and that's your water. And that leaves these two ends open, and they can link together to form your compound. So we're forming this compound, synthesizing this compound, by removing water from the reactants, by dehydrating the reactants. There goes your water off into solution. Okay? The opposite of that is we're taking our compound here, and there it is again, and in order to break it apart, we're going to break this bond. We could break it there too, doesn't matter. But in order to break this bond, we're going to add water back into the solution. And when that happens, this bond is broken. That's the lysis part. And water dissociates into its component ions. Remember, water is made up of a hydrogen ion plus and a negative hydroxyl or OH ion. And so those guys tag on to these molecules, the opposite size obviously, to complete the structure, right? Because otherwise we'd have two open bonds here. We don't want that. And so these two ions of water make their way to cover those bonds, and now we have our original reactants back here, simply by adding water. So we break this bond, we lyse this bond by adding water. Hydrolysis, hydrolysis. We'll see that over and over and over again. It's a running theme here. Alright, so we want to make carbohydrates now. So carbohydrates, remember, are chains of glucose. They're used in organic systems for storing energy, as well as structural components of things like plant cell walls and arthropod skeletons. So carbohydrates are polymers, remember that word, of simple sugar monomers, namely glucose, but that's not the only simple sugar, but that's the one that we're going to be dealing with right here. The chemical formula for glucose is C6H12O6, and that's a simple sugar, and we'll look at it in a minute, its structure in a minute. And so carbohydrates are formed by linking monomers or glucose units. And how do we do that? Dehydration synthesis, right? We already talked about that. We're going to manufacture something. We're going to synthesize something by removing water. So we have a glucose molecule on one side with an OH, a glucose molecule on the other side with an H. We pull the OH off, there goes your water, and now those two glucose monomers can link together. And now we have a disaccharide, we call it. Glucose would be a monosaccharide, a single sugar unit. Now we have two glucose molecules hooked together, so now we have a disaccharide or a double sugar. Right, so two units of monomer are joined together by removing a water molecule between them, and the resulting chemical bond is a glycosidic bond. Important that you remember the names of these bonds for all of these different categories of macromolecules. So we're going to build carbohydrates, large carbohydrates, via dehydration synthesis, link them together by removing water, and that bond that results from that is called a glycosidic bond. And here we go. So here we have two monosaccharides or monomers of glucose. See? And each of them has a little OH hanging off. So what we're going to do is we're going to pull the OH from one, and we're going to pull an H off of the other. That's your water. They link together, and that's your water, and there it goes. And now we have an open situation here. So now these two can link together via that free oxygen, and now we have this disaccharide, this double sugar, this sugar made of 2 units of glucose. By the way, it's called maltose. All right, but this is your glycosidic bond right there. Bond between 2 monosaccharides. Right? Easy enough. How do you do it? Remove water. Dehydrate the things. What if we wanted to break this apart? What if we wanted to denature this disaccharide? We want to break it back into glucose monomers. Well, we do the reverse. We add water. That pops this glycosidic bond open. The OH of one, the OH ion of one will go to one molecule of glucose, and the H ion, the hydrogen ion, will go to the other monomer of glucose. And then we have our glucose back. Okay, proteins. Proteins are used in organic systems as functional structural elements, which we'll see. Functionally, proteins serve as enzymes. Enzyme is just a word that means organic catalyst. All right, enzyme. It's a catalyst. What do you know about catalysts? Anything? Think about it for a second. Catalysts, or a catalyst, is added to a system to speed up the reaction of that system, right? To speed it up. So in the case of the space shuttle, for instance, right, there is liquid fuel which will make the thing fly. But they put liquid oxygen, they mix liquid oxygen with the fuel to catalyze the burning of the fuel to give you more power and more thrust, and so the thing can fly into space. Not a great example, but it's the same idea. So an enzyme is a protein that catalyzes a reaction or speeds up the reaction. We'll see the importance of that in a little while. Proteins are also used as transporter molecules like hemoglobin. Your book uses the example of insulin. Hemoglobin transports oxygen and carbon dioxide in the body. And proteins are also used structurally with things like hair and silk. Okay, proteins are long polymers, just like carbohydrates are long polymers. Proteins are long polymers of amino acids. Let's check them out. So proteins are formed by the linking of monomers of amino acids. You know what that is now, right? An amino group on one end, a carboxyl group on the other end, positive, negative, it's polar. Via how do you link things together in organic systems? Sounds like a broken record. Dehydration and synthesis. Hook them together by removing water between them. So 2 units of amino acid are joined together by removing a water molecule, that's H2O, between them. I've written it like this to remind you that the OH ion of water comes from one side of the reaction, or what's going to be the linkage, the bond, and the O really should be an H over here, it's not HO2, it's H2O, so this should be an H, and the H, the hydrogen ion, comes from the other side, comes from the other element, comes from the other monomer, okay? Sorry about that. This should be OH and H to make up water. So the resulting chemical bond is called a peptide bond. I explained this a little while longer. We'll see it a little while ago. We'll see it again in just a minute. Peptide bond between amino acids. Proteins are also known as polypeptides because proteins, as I've said, are big long chains of amino acids. Right? And so if you have 1, that's a monopeptide. If you have 2, that's a dipeptide. If you have 3, that's a tripeptide. If you have 4 or more, we call it a polypeptide. And it could be 4, it could be 104, it could be 4,000. Big long chains of amino acids. And here we go. So peptide bonds. So the covalent bond between the amine, or the amino group of one amino acid, and the carboxyl group of another amino acid, we call it a peptide bond. And in order to get there, you have to remove water. This is dehydration synthesis all over again. So we pull the OH off of one amino acid, one end of one amino acid, and we pull the H off of the other end, the opposite end of another amino acid, an adjacent amino acid. That's your H2O, there's your water, and there it goes. And now we can hook these together because we have an open linkage there. We can hook them together, and that's your peptide bond. Remember, peptide bond, the carboxyl group of one amino acid binds to the amino group of an adjacent amino acid. And remember, it doesn't show it here, but the carboxyl end has a net negative charge. The amino end has a net positive charge, and so this guy right here is polar. And that's your peptide bond between amino acids. Okay, so synthesizing protein. Okay, I showed you this already. It's the same situation. Just a long chain, a protein, long chain of amino acids. Here are your peptide bonds. Carboxyl group of one binding to the amino group of another. That is your peptide bond. You have many peptide bonds in here, and that's why we call it a polypeptide. And remember always, when it comes to proteins, that the N terminus is positive, net positive, the C terminus is net negative, and so proteins are polar. That's important to remember when we start talking about DNA and RNA, okay? I just made little notes down here. A dipeptide is 2 amino acids, a tripeptide is 3 amino acids. If you have 4 or more, we just call it a polypeptide. Poly means many. And again, this could be 4, 104, 400, 4,000. Okay, it's a lot. It can be a lot, and in most cases, it is a lot. Okay, lipids. Lipids are used in organic systems for storing energy, as well as structural components like cell membranes. We'll talk specifically and extensively about phospholipids. By the way, did you know all of the membranes surrounding your cells contain cholesterol as well? Steroid hormones are lipids. Lipids include fats and oils like butter, corn oil, soap, and wax. And lipids, I think you know this already, are insoluble in water. In other words, they do not dissolve in water, right? Oil and vinegar, right? I used this before. Vinegar is an aqueous solution. Your oil does not mix up with it. So in order to put it on your salad and enjoy it, you shake it together. It still doesn't mix together, but you have globules of oil floating around in the solution of vinegar, and you get the flavor of both on your salad. Okay. Lipids, insoluble in water, and think of fats and oils. You can't dissolve them in water. Okay, synthesizing lipids. Lipids vary according to chemical structure. We have triglycerides, phospholipids, steroids in the case of hormones. So let's talk about triglycerides real quick. Triglycerides. These are, so you have three components, three subunits connected to a glycerol molecule. Glycerol is just a little three hydrocarbon chain. So triglycerides, and you're familiar with that term, are formed by linking three fatty acids. That's your monomer subunit, three fatty acids, to a molecule of glycerol. And again, how do you hook the fatty acids onto the glycerol molecule? Think about it. How do you do it? Synthesis, dehydration, synthesis. You remove water. Let's look at it. So formation of triglycerides. Here's your little glycerol molecule. It's just a little hydrocarbon chain. Three little carbons there. Each carbon has hydroxyl group attached, an OH attached. Here you have three separate fatty acid chains. We call these hydrocarbon chains. Just carbon and hydrogen, carbon and hydrogen, carbon and hydrogen, carbon and hydrogen, over and over again. But however, on one end there is this carboxyl group, right? The carbonyl group, double bond, and the OH hydroxyl group attached. So in order to attach this chain to this glycerol molecule, we're synthesizing it, we've got to remove water. So there's your OH there coming from your fatty acid, and your H there coming from your glycerol. Take them away, there goes your water. And now these two can hook together. But this is a triglyceride, so you have to do it 3 times. So that means you're going to remove ultimately 3 molecules of water, because you got to do this 1 time, 2 times, 3 times. So you have to do a dehydration synthesis 3 times. It happens all at once, but the point here is you do it, it happens 3 times, because you got to get rid of 3 molecules of water. There they are over there. And here's your triglyceride. Your glycerol molecule connected to 3 fatty acid hydrocarbon chains. So another example of lipid is the phospholipid. By definition, it's a fatty acid chain with a phospho, meaning phosphate group attached. So phospholipids are formed by linking 2 fatty acids, which is your hydrocarbon chains, plus a phosphate group to, once again, a glycerol molecule. How do you hook them together? How do you synthesize anything in organic systems? Dehydration synthesis. Remove water. So here is the phosphate group. Phosphate is a phosphorus group, or a phosphorus molecule, linked to 1, 2, 3, 4 oxygens. Right? This is your R group, your variable group. But that's the phosphate group. A phosphorus linked to 4 oxygens, okay? And we're going to hook that up to 2 fatty acid chains. And that's going to make a phospholipid. Phosphate and lipids, phospholipid. So looks like this, synthesizing phospholipids. Here is your phosphate group. Here is your little 3-carbon glyceride or glycerol group. And so the phosphate hooks on one end of the glycerol, and then here are your fatty acid side chains, right? And what we're doing here is we're removing, you don't see it here. Not a good picture, but I'll try and get you a better one. But you don't see it here. You're removing an H2O here, and you're removing an H2O there, right? And you're removing an H2O there. That's how you get these things hooked up. Dehydration synthesis, right? But your phospholipid, the point here is the phospholipid is a glycerol molecule, 3 little carbons, attached to 2 fatty acid chains, or lipid chains, lipid carbohydrate chains, hydrocarbon chains, and a phosphate group. And this is really important when we talk about cell membranes. All right, nucleic acids is our final one here before I end this first part. Nucleic acids are DNA and RNA. All right, when you think of nucleic acids, think DNA and RNA. Nucleic acids are used in organic systems for encoding genetic material. We know them as DNA. RNA, by the way, that's deoxyribonucleic acid, DNA, and that's ribonucleic acid. All right? So nucleic acids are formed by linking nucleotides, that's the monomer subunit, via, how do you hook things together in organic systems? Dehydration synthesis. Take out water, create a bond, boom, you got it. So nucleotide is a molecule of a pentose sugar, we'll look at that in a minute, the structure, linked to a phosphate group, we've seen that already, and a nitrogenous base. We'll look at the structure here in a second. And there you go. Here's your pentose sugar. Pento, penta. The root word there is pent, and it means 5. So pentose sugar, we see 1, 2, 3, 4, 5 sides, right? I mean, it's drawn out like this. Looks like the pentagon, right? That's where pentagon comes from. It's a 4-sided figure. But really, we're talking about the carbons here. There's a carbon here. We'll sit up close in a minute. There's a carbon at each corner. Well, except that one. That O represents oxygen. But there's a carbon at each corner. Carbon, carbon 2, carbon 3, carbon 4, and then the 5th carbon is hanging off here. Right? Like that guy's out there just waving. So 5 carbons. That's the pent part. And this happens to be a sugar molecule, right? This is a monosaccharide, if you will. And up here on the 5th carbon is attached, look there, a phosphate group. A phosphorus with its 4 oxygens. And then over here, off of carbon 1, is attached the nitrogenous base. Nitrogenous bases are these ringed structures, and we'll look at them in a minute. There are 4 of them. Actually, there are 5 of them. However, 2 of them are interchangeable. But a nitrogenous base, why do they call it a nitrogenous base? Well, look at all the nitrogens in there. Right? Look at all the nitrogens. Where this thing has carbons at the corners, and then one out there, this thing has nitrogens at many of the corners, and then one out there. So that's where it gets that's where you get the term nitrogenous base. So nucleotide, nitrogenous base, pentose sugar, phosphate. So dehydration synthesis links two nucleotides together. So when we are talking about making, remember, nucleotides, or nucleic acids rather, are DNA and RNA. DNA and RNA are made up of chains of nucleotides. Nucleotide is the monomer. So how do you link two nucleotides together to eventually get DNA or RNA? Ding, 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 dehydration synthesis, see? How do you get them apart? Hydrolysis, hydrolysis. So we hook them together by taking away water, we break them apart by adding water. Let's look at it down here. So here we want to put, here is your nitrogenous base, your pentose sugar, and your phosphate group of 1 nucleotide. Here is your nitrogenous base. Notice this is a double ring structure. This is a single ring structure. Your pentose sugar and your phosphate of another nucleotide. And look what happens. The hydroxyl group on the number 3 carbon of 1 nucleotide gets linked up to the phosphate group of the adjacent nucleotide by the removal of water. There's your OH, there's your H, your H2O, boom, there goes your water. And the bond is formed between the oxygen here on carbon 3 and the phosphate group. You want to break them apart, you want to get them apart, break that bond right there. By the way, that's a phosphodiester bond. We're going to break that bond. How do we do it? Hydrolysis. We're going to lyse the bond by adding water. Here's your water coming in, hydrolysis or hydrolysis, and boom, now we go back to our original 2 nucleotides. We'll look at this playbook over and over and over again. Okay, synthesizing nucleic acids. We're talking about nucleic acids again still. The sugar element of nucleic acid is a pentose sugar. Pentose, as I've explained, is a 5-carbon sugar in a ring formation. If you draw it out on paper, it looks like the pentagon. So, RNA stands for, it's an acronym that stands for ribonucleic acid. The sugar that we're talking about is ribose, R-I-B-O-S-E. That's the R in RNA. It stands for ribose. It's a pentose sugar called ribose. We'll look at its structure in a second, although you've seen it already. And if you're familiar with DNA, you know that as deoxyribose nucleic acid. So it's like saying deoxyRNA. How does that happen? We're going to deoxy RNA. This is ribonucleic acid. This is deoxyribonucleic acid. So we're going to deoxy the ribose sugar in RNA. That's how we come up with deoxy in DNA. So the pentose sugar, still a 5-carbon sugar, is called deoxyribose because we're going to remove, guess what? An oxygen. How do we do that? So here's the generic structure of a pentose sugar. This happens to be, this is ribose. Here's your 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon hanging out there. Hey! And you'll notice that carbons 1, 2, 3, and 5 have OHs, hydroxyl groups attached to them. So when we think of ribose, all of these hydroxyl groups are in place. All right, so this would be an example of ribose. Now, if we want to make deoxyribose, we want a pentose sugar that's deoxygenated. We're going to take this ribose and we're going to remove that oxygen right there. Get rid of the oxygen. We've officially deoxygenated ribose, and now we have deoxyribose. So the nitrogenous base element of a nucleotide is one of 4 nitrogen-containing ring or double ring molecules. Okay, now as we get into in the second half of this chapter, building DNA and RNA, it's important to remember the nitrogenous bases. So DNA, this is deoxyribonucleic acid, contains deoxyribose as its sugar. Has the bases adenine, we denote that with a capital A, thymine, that's a capital T, cytosine, that's a capital C, and guanine, that's a capital G. Just take these as they are right now. We'll learn the details and why those letters are important later on. And then ribonucleic acid contains the sugar ribose, and it contains the bases adenine, capital A. Instead of thymine, RNA contains uracil, and that's the exchange that's made between DNA and RNA. So when you go from DNA to RNA, instead of translating thymine, it's going to translate as uracil. That's a capital U. And cytosine and guanine are still the same. Okay, so first let's look over here, DNA and RNA. Now, DNA, as we'll learn, is a double-stranded molecule. That's what these little two, it looks like a little twisted ladder. That's we call it the double helix. RNA is a single strand, or a single-stranded molecule. RNA has ribose, the sugar ribose is a pentose sugar, and all of the little OHs are in place. Oh, there they are. There's our little OHs, little hydroxyl groups. And DNA has deoxyribose. We're going to deoxygenate ribose, get rid of that oxygen on carbon 2, so that this becomes deoxyribose. Okay. Here are your bases over here, and we call so this says sugar phosphate backbone. Right, so when you see a molecule of DNA or RNA drawn out like this, these wide lines represent this sugar phosphate backbone. That's the pentose sugar connected to its phosphate group, and that never changes. Okay, so whether you're talking about DNA or RNA, that's not going to change. What does change are the bases out here. Remember, the bases are attached to carbon number 1 of the sugar. Those are phosphodiester bonds. I think I mentioned that. And so look at the structures here. Thymine T, right, is a single ring. Adenine here, that's A, is a double ring. And that's really important when we think about building DNA. Structurally, we're going to see a double ring connect to a single ring. You're never going to see 2 single rings connected together. You're never going to see 2 double rings connected together. It's always double single. So in other words, in the case of DNA, thymine is always going to link up to adenine. Down here we have cytosine. It's another single ring structure. We call these purines. Purines, double rings are pyrimidines. And here's guanine, another double ringed structure. And guanine always binds to cytosine. And so we'll see, you'll hear later on me referring to A binds to T and G binds to C. In the case of DNA, and the change here comes in the T with RNA. T becomes uracil, which is obviously is going to be another single ring structure. All right, so that concludes this first part of Chapter 3. We're going to talk about more about phospholipids and more about DNA and building DNA and transcribing RNA from DNA and translating RNA into protein to eventually get to that big long chain of amino acids, that polypeptide. That's kind of where we're going here, right? And we'll see that in the latter half of Chapter 3. If you're reading ahead, or if you have read ahead, then you've already been exposed to that. So I will see you next time for Part 2 of Chapter 3, Biological Macromolecules.